Okay, so I will guide you around the website, and I will show you the capabilities, the content, and some examples of how it can be used in research on on anti-cancer drugs. And just to start, DrugCARD is a cancer therapeutics and drug database, which has been designed for researchers, clinicians, and developers of anti-cancer therapies. So it will allow users to search in the resource for anti-cancer drugs, compare them, compare their usage, regimens, and also combinations used in clinical, but also combinations used in preclinical studies. And for example, assess how drugs can be combined according to their mechanism of action, their targets, the tumor types, if drug A should be given before drug B, or at the same time, or B before drug A, and so on, how they can be synergistic, antagonistic, etc. So DrugCARD currently contains more than 160 compounds, which are used in the clinic or are still in development. And it allows searching using various criteria and categories. So you can search drug by name, but also you can search for tumor types. We have currently 39 tumor types, 25 targets, also called mechanism of action for the drugs. And the content for each drug which is given is modeling data. So you will have data parameters for pharmacokinetic and pharmacodynamic modeling, preclinical combination and sequence outcomes for a selected number of compounds, 700 regimens used in the clinic, regulatory agency label outcomes, and literature references. So I will start by the beginning, which is really that you need to create an account to access the database. So you have the possibility to directly click on register on the left here, and then enter your details, classic thing. Or you can also create an account as a free trial. We offer currently a 14-day free trial for DrugCARD with full access. So you can enter a username, choose a password, put your details if you work for the industry or academic. We do not require any billing details for a free trial account. However, you are free to enter them and then to extend your account to a full registered account. A year account costs £1,000 per account in the industry and £200 for academics and non-profit organizations. So just a note, we do not keep any billing information on our servers. So payment processing is done through a certified payment processing company, and your details are not stored, such as credit card details, for example. But let's start with the science now and how you can use the database. So I will connect now. And as you can see, when you are connected, you can use the user panel on the left. If you click on your name, then you are directed to a user panel where you can do classical things with the user panel, which is changing your passwords, update your personal details, update your billing information, and if you wish, extend your subscription. But let's go back to the search now. And as you can see, the search panel contains 4 categories: drugs, tumors, regimens, and targets. However, they are all linked. So, for example, when you will search for a drug and find records for a drug, a lot of the data concerns tumor types, a lot of the data will be regimens, and also mechanism of action for the drug. So each name which is referenced here in the 4 category are hyperlinked, and you can go back and forth from one category to another just by clicking. I will show that to you in a second. But let's start with searching a drug by name. So that's very simple. You have a search input where you can put a name, for example, docetaxel. And I can search, and then it returns a hit, which is here docetaxel. And it gives also some alternate names, which can be other generic names, USAN, non-proprietary names, also commercial names. So for example, if I type Tarceva, then I will find erlotinib. The name which is linked is always a non-proprietary name, but you can search indifferently erlotinib or Tarceva. You don't need to even search for the full name of your drug. So for example, gemcitabine, you can type only GemC. If I search, I will find gemcitabine, also called Gemzar in the commerce. If I put only gem, then I find these two drugs, gemcitabine and gemtuzumab. So now, when you want to have details on the drug you search for, then you just click on the hyperlink. And this is the record that you get, which is organized as cards with relevant information which is contained in collapsible panels. So as you can see here, for gemcitabine, we have here a drug name, gemcitabine, with a trade name of Gemzar, and different panels that contains information on targets or mechanism of action, pharmacokinetics, pharmacodynamics, outcomes, dosing and labeling, clinical regimens, combinations, and references. Just before going into the detail of the information which is contained in the database, you will notice that each collapsible panel has a light border, a colored border. And the color corresponds to the kind of data that you can expect inside each category. So blue is for general information for the drug. So you will have here the names, trade names, and at the bottom the references where the data which is shown above is extracted. Red is for clinical data. So that's data that has been determined in the clinic in human. And green here for combinations for preclinical data. So the combination here are given in preclinical experiments such as xenograft or in vitro cell culture. So each panel, as I said, is collapsible, so you just need to click on it and then it expands and shows the detail. If you click again, it will collapse. And let's start with the beginning, which is, for instance, the targets. So each target is a mechanism of action. So here, gemcitabine, for instance, has an S-phase specific cytotoxicity. If I click on the S-phase specific cytotoxicity subpanel, then it expands as well, and you can see the data which is there. Species here is human, obviously, because it's a red border, so clinical. And then you will find some data for modelers, such as a dose response equation. If you click on the little question mark here, you will be directed to a page with different dose response equations that we use in the database. So most of them are hyperbolic dose response model, also called Hill equation, the very famous Hill equation. But we can use also 4-parameter logistic and logarithmic dose response model. So if I go back to the gemcitabine, as you can see, you can find values for inhibition of a target. Here is the S-phase specific cytotoxicity. And if you click on target details here, it will go to the mechanism of action and with the different drugs which are referenced with equations. In pharmacokinetics, you will find also clinical data for different parameters for the PK modeling. So you will have elimination phase half-life, alpha phase half-life, and intercept, volume of distribution, all bioavailability, and rate constant overall absorption. Pharmacodynamics contains data on immunosuppression, mutagenicity, mutation rate per cell division, reversion rate, and gross log kill at the maximum tolerated dose. All this data is extracted from the references, which is given on the reference panel here. Outcomes are regulatory agency labelings. So at the moment, we have FDA labels, but we are in the process of adding EMA labeling as well. Dosing information gives you the maximum tolerated plasma concentration, maximum tolerated cumulative dose, and maximum tolerated single dose here. Finally, clinical regimens are regimens used in the clinic and involving here gemcitabine. So, for instance, the first one here is given here. On the right, you have a type of cancer. So again, if I click on the cancer type, then I will be directed to the bladder cancer page, and you can see all the clinical regimens used in bladder cancer here, and also the information on combinations, preclinical combinations. It's a green panel using gemcitabine, using all kind of drugs actually in bladder cancer. Going back to where we were with gemcitabine. And if you click on details on the regimen, then you will be given the full regimen details with the references where the regimen has been published. So the regimen contains the information on dosing, route, and also on which days the drug is given and how many cycles the drug is given, and so on. Combinations are preclinical combinations, and many drugs have been combined. And in preclinical, a lot of data has been given on how synergistic, antagonistic, or additive they could be. And very often that synergy is dependent on the sequence of the drug. So sometimes it's better to give drug A before drug B, and instead of drug B before drug A. So that's where you have here, for example, here it's a combination involving gemcitabine, but 5-fluorouracil was given before gemcitabine. The cell line is a colorectal cancer, it's HT29, and we give the outcome. If you click on the link here, then it will download the paper, depending if you have access to the editor's paper. If the paper is free, it will be downloaded directly. So another example here is gemcitabine followed by irinotecan. It's pancreatic cancer cell line, BXPC3. Here we give the timings. Gemcitabine was given for 24 hours, then the cells were washed, and irinotecan were given for 24 hours. And the outcome was that that combination was synergistic. Again, you can find the paper by clicking on the link given there. So the idea is to provide researchers a tool that give them an idea on how drugs can be combined, how they are synergistic, how it can help with designing new experiments, administration, scheduling. And just one more word about the search. Here I showed how we could search by name. However, if you are interested in just looking into a list, it's still possible. So I'm going back to the drug search information page. And under this input box where you can search, you can simply click on the link given below. Click here for a list of all drugs. And here you can see all the drugs that are in the database. So drugs can be small molecule, biotherapies, large molecule targeted agents, all kind of anti-cancer drugs. For tumors, it's the same. You can search for a tumor name. So bladder cancer, for example. But if I click here for a list of all tumors, then you will see the 29 different tumor types that we have in the database. Regimens, it's the same. We have more than 700 regimens. And for targets, we have we have 25 targets or mechanism of actions. So as you can see, from angiogenesis inhibition or RA kinase, c-Abl, to specific cytotoxicity, phase-specific cytotoxicity. So at the moment, the data has been curated from literature, and we are constantly extending it with new information as it comes. We have also in the future plans to provide with statistical tools that will give you, for example, when you search for a drug, the percentage of regimens where it has been combined, with which drug it has been combined, and so on. You can look at the user guide that gives you an idea on how to use the DrugCARD. I hope that this demonstration will show you that you don't really need a user guide to use. You can use also the contact us page to tell us about any error that you could see, any bug that you could see, but hopefully you won't find a lot. So you will use rather that feedback form to let us know which features you would like to see added to DrugCARD. And if you have other issues with the registration, do not hesitate to contact us. So that's it. And now I think we can start a question and answer session. And I thank you very much for attending this session. And I will be pleased to answer all your questions now. And if you don't have time, then do not hesitate to send me an email, efernandez@physiomics-plc.com. I will be pleased to assist you and to answer any of your questions. Thank you very much.